very useful definition because it means basically nothing. It just means something that is really, really small. The thing is, however, it is useful because if you think back, if it's too small to be seen with the naked eye, we have no idea that it's there. And that's what people used to think before the invention of the microscope. People thought that disease was an act of God, or was carried over with the wind, kind of right on that part. And it just happened to bad people to get disease. That's fortunately not quite true. In 1665, Robert Koch, he had the first kind of, and we talk about microscope more next time, he was the one who had the first rudimentary microscope. So he looked at, used that thing, and he saw something that would later be called cells, little tiny structures, the tiniest little structures. It really started taking off with Leeuwenhoek in the 1600s and the later part of the 1600s, because he actually had a much better microscope at hand, and he could actually see moving things in his slides. He called them animalcules because they were running around on his slides and moving. So these were the first microbes, the first bacteria that he saw, probably also some amoeba. He used a really, really good magnifying lens for that, to see those. He was probably the best lens maker of his time. So that's really what started the study of microbiology. And that kind of settled the question of whether, or started the question whether disease or animalcules derive spontaneously, or generate spontaneously, or whether they're come from something that was there before. So the spontaneous generation idea is, the spontaneous generation hypothesis is the idea that life arises from non-living matter, rocks, stones. Right now we laugh about it. And that a vital force, a life force, has to imbue these materials to actually make them into these small living things. However, the conflicting view is that it's biogenesis. Living things can only arise from other living things. How do we test it? Well, here's a very quick overview of a couple of experiments people did. Already you filled jars with decaying meat. And what happens? Well, if you covered the decaying meat with fine nets, you got no maggots. So no life force. However, if you have open jars, maggots appear. You seal the jars, again, no maggots. So where and how did the maggots come from? Is that life force that used the decaying meat to make it into maggots? Or is it something that seeps into the jars from the outside? Anita did a similar experiment. 100 years later, he put nutrient broth, really rich nutrient broth, into covered flasks and boiled those flasks. And lo and behold, if the nutrient broth is heated and then placed in the covered flask, microbial growth. Where does that come from? Again, is it generated or is it spontaneous? Spallanzani, he boiled the same nutrient broth, however now in a sealed flask. Oh, he does not get microbial growth. So already the thinking goes towards, well, it's spontaneous generation may not be the thing. If you seal a flask, whenever people do these sealed experiments, no growth occurs. And then finally, Louis Pasteur, he really put the nail in the coffin of the spontaneous generation idea. It took another 100 years and a lot of chemical expertise to do that. He did a very famous experiment with these S-shaped flasks. So what he did was he had a sterile flask. Back here, he used an S-shaped flask, kept broth in it, and he put a liquid in that swan's neck of that flask. That means microbes cannot go in, but air can go in. So when he does that, the broth in these flasks shows no sign of life because the neck of the flask here would trap spontaneously just rip the neck off, open up the flask to the outside, and now all of a sudden you would see growth in that flask. That really demonstrates that the very same broth, under two different conditions, whether it's open or not, either has growth or no growth. So the growth in there must come through airborne microbes that infiltrate this thing. So spontaneous generation is completely dead after this experiment. It's biogenesis, growth, life can only come from pre-existing life. In fact, one of his flasks, an unbroken flask, still with liquid in the S-neck here, is still on display in Paris 150 years later and still shows no sign of growth. That's really an ongoing experiment, and it confirms every day that the biogenesis idea of microbial growth is the one that is true. And here's just another picture of that experiment. So that directly leads to germ theory of disease, that microbes that are in the air, in the soil, everywhere can cause disease. And that disease is nothing that is bad wind, bad faith, whatever. It's really brought about by these small little microbes that are everywhere in your surroundings, and they can cause the disease that you have. If you have that, if you work on a germ theory, you also can develop very easy ways to counter microbes and disease. And guess what happens? That's through the use of aseptic techniques, sterilizations, hand washing to really kill those microbes. That was the single most important factor in increasing our life expectancy from, say, 60 years to what we currently have, which is 85 years. At the turn of the last century, population growth was basically a flat line like so. But as soon as people realized that the germ theory was the one to look out for, and people started using aseptic techniques in hospitals and at home, population growth went exponential. And now we have 8 or 9 billion people on this planet. Back in 1900, it was maybe 1 billion or so. And the single most important factor is antimicrobial medicine. Okay. We don't need to talk about it. So what are microbes then? Microbes can be all sorts of different things. It can be bacteria, it can be spores, can be amoeba, can be viruses, can be algae. Just a couple of pictures of those. In order to classify them, we can talk about bacteria or prokaryotes, which have a cell wall, no membrane-bound organelles, and a cell wall that is made from peptidoglycans. Archaea are a really strange form of bacteria, so strange that they constitute now their own kingdom or domain of life. They are also prokaryotes, but they have a cell wall with no peptidoglycans. And when we talk about prokaryotes and eukaryotes, what the cells are made of, that will make a little bit more sense. Eukaryotes are what you are, that are cells that contain membrane-bound organelles like mitochondrion, like nucleus, and so on. In eukaryotes, you usually differentiate 4 larger groups: the fungi, the protists, the plants, and the animals. We are here all animals. Protists, that may or may not be a true group. Basically, what people grouped under protists was a big conglomerate of things that they had no idea what to do with them. So amoeba feature under protists. Algae feature under protists. So they have really nothing in common with each other. That's why the group of protists is not really considered seriously a viable group anymore. Other types of microbes are viruses. They may not even be alive. If life is defined by reproduction or by copying yourself, viruses can't do that. Viruses need a host cell to copy themselves. They don't belong to any of these major groups of microbes because they're technically not alive. Yet they reproduce and they're massive sources of And then we have something that I just really just added to the top, prions. Have you heard of prions?
No.
Anyone heard of prions? Have you heard of mad cow disease?
Yeah.
Yeah. Have you heard of scrapie, kuru? These are all prion diseases. And mad cow disease is a fantastic name for that disease because it literally melts your brain and makes these cows act all crazy like. Human versions of that are Creutzfeldt-Jakob's disease, or kuru, which is endemic among cannibalistic tribes in Indonesia. Prions are something that completely defies expectation because they're not cells, they're not cell-like. At least in the virus, you see elaborate structures. You have an outside, there's genetic material on the inside. Prions are just very, very small proteins, like egg whites. That's how they look like. And they can replicate themselves, but they can do anything else. So they're the opposite of the virus. The virus can do a lot of things except replicate itself. Prion can do nothing, but replicates itself very, very efficiently. And that's why it's infectious, and as a consequence of its own self-replication, it literally melts brains and punches holes into people's brains. And if you're a cow, then you go mad, you can't walk anymore, eventually you fall down, and you have to be euthanized. Humans, similar things happen. They were discovered about 20 years ago. They must have been around for a really long time, but we had just no idea about this. This is a completely nonsensical branch of microbes. People really don't understand them because they're just so bizarre. Okay, we have about 10 more minutes. Quickly, bacteria are prokaryotes. Prokaryotes use pre-nucleus. They're single cells. They have peptidoglycan cell wall, and they're reproduced by basically dividing itself in two. We'll have sessions on bacteria and eukaryotes, so we really don't need to go into much detail right now. Just very quick overview of microbes that you'll encounter. Archaea are prokaryotes, but they lack peptidoglycan in their cell wall, and they live. Their claim to fame is that they live in really extreme environments. They live in hot springs where the water temperature is near boiling. They live at the bottom of the ocean where volcanoes erupt into the ocean. They live in salt that is almost so salty that it's solid. Great Salt Lake in Utah or the Dead Sea, that's where halophiles live. So these are characterized by the love of extremes, and people think that these were amongst the first living things on this planet, because when the planet formed, temperature, salt, and whatever concentrations were just really extreme. And that's where the archaea come from. Fungi are eukaryotes, so they have a distinct nucleus and organelles within them. The interesting thing about fungi is that they have chitin as a cell wall. Chitin is the same thing that makes the the insect exoskeleton. So they're a weird combination of insect and cells. When they're big, they become mushrooms, but those are multicellular ones, so they're a little bit slightly different. Protozoa, or amoeba, these are eukaryotes, and they are basically little crawlies, creepy crawlies. They walk around surfaces with pseudopods, basically their own little version of feet. Pods mean feet. So they extend little parts of the cell membrane to walk from one surface to the next. In fact, you have sort of like amoebas in your blood. Your macrophages of your immune system do the same thing. They walk around in your blood vessels on the hunt for foreign objects, including bacteria and viruses that infest your body. Finally, algae, they live in the water. Their claim to fame is that they are photosynthetic. So mostly they're green or red, so that they can absorb sunlight, and they use the sunlight to make their own energy, just like plants do. In fact, algae are thought often to be precursors of plants because of their photosynthetic ability. Finally, viruses. As I mentioned before, they're acellular. They don't even have real cells. They have DNA or RNA as genetic material, as all cellular things have. But they're really just a surrounding, they have a protein core around that DNA, and that's it. So they don't have any of the tools that the cell has to live, to make energy, to reproduce, to create more of its cells. They just have DNA and a little bit of protective shell around it. And in fact, they hijack other cells to do the dirty work for them to reproduce. So a lot of people, or the discussion, the debate is still, is this even alive? We don't even know whether that can be considered alive or not. And then finally, prions, I don't have them here. Prions are simply protein, even more bare bones than the viruses, and yet they reproduce like crazy. So you'll meet a lot of really bizarre organisms during this course. Multicellular parasites are mostly animal-like. Eukaryotes, they could be worms, flatworms. You may have heard of a tardigrade, which is one of those animals that actually survives in space, that lives on your eyelashes mostly. And you can send it out into space, send it out in the vacuum of space where it's minus 200 Kelvin temperature, where there's virtually no air, where there's an absolute vacuum, and they come back, you put a little bit of water on them, they're just happy. So the relationship between all those, I talked about there's three major domains of life, which are the eukarya, the bacteria, the prokarya, and the archaea. And every life, as you probably know it, mostly consists of eukaryotes. That's where the animals are, the plants are, everything that you know that you interact with is probably in the branch of eukarya. However, nomenclature. Just want to point that out. However, even though you're a eukaryote, you're definitely not alone in your body. The amount of bacteria in your body, on your body, there are more cells, or as many bacterial cells in your body than there are eukaryotic cells in your body. By weight, you're only 50% of you. The number used to be 10 times higher. They estimated it to be 10 times higher, that you're only 10% you, based on cell numbers. They measured that just in January this year. Now it's a 1-to-1 ratio. So taking over a little bit. Taking some of the terrain back. We're a little bit more human now than we were before. All right. Just one more quick thing about nomenclature, and then we can take that break. The nomenclature is called the binomial nomenclature because every organism has two names to its thing. It was established by Linnaeus, who is the godfather of taxonomy. He started out in 1700. He went around and gave everything that he saw names, classified it based on its observable features into groups, and he had to name them. He had to make a nomenclature for that. So basically, each organism has two names: the genus and the epithet. What does that mean? The genus is the first name. Usually is that describes the whole class of organisms that belong to that very same genus. The epithet is something that is being added on to the back of that name to really highlight this particular organism. And that can be anything. The genus has to be scientific based on where you find that organism. The epithet is something that you can make up. A lot of famous scientists have organisms named after them in the epithet of that animal or that organism. And since it was developed in the 1700s, Escherichia coli is named after Theodore Escherich, who discovered these bacteria. These are the bacteria that live in your gut. It's called coli because it lives in the colon, in your gut. Staphylococcus aureus, it describes the shape of that thing. These are cocci, small round, cocci are small round cell, staphylococci are spherical cells. So this is merely a descriptive name. These are staphylococci, and because it's gold looking, they use the Latin word for gold. So these are just a couple of examples. You'll learn, you'll see this nomenclature a lot throughout this course. That's why I'm just throwing this out here. The way we can talk about this, we don't need to use the full name. Just abbreviate it.